Hey guys, what's up? So I told you guys that I would be posting up a video going over the recombinant DNA FRQ. That was the one that was really difficult. So what we're going to do, we're going to go ahead and go over that today. That way you can go through, you can check your answer. I'll try to provide feedback. I'm getting a little bit behind on grades just because Miss Rodriguez, as you know, already had her 2 babies. But Ms. Villarreal also had her baby this weekend, so I'm trying to catch up with all 3 sets of classes for their grades. So I did not forget about you guys. I could never forget about you guys. I'm going to go ahead and I'll start providing feedback. That way you can, work on answering those FRQs. Until then, we're also going to be doing some ed puzzles now that College Board has come out with a few more videos. So that way you can review some older material too. So let's go ahead and get started. All right, so I have your recombinant DNA FRQ. So this one was a lot longer. It's worth 10 points. So I'm going to go ahead and read through this prompt, and as I'm reading through, I'm going to go ahead and underline or highlight things that are important. That way we need, to just be able to pay attention. All right, so CD3 is a signaling protein that is typically found only in the plasma membrane of immune system T lymphocytes. CD3 is composed of several different polypeptides, including a gamma chain, CD3 gamma. Scientists analyzed the promoter of the CD3 gamma chain, let's see here, gene, for regulatory sequences that might have positive or negative effects on the expression of that gene. Okay, so we haven't talked that much about positive or negative effects, but you did talk about that quite a bit in freshman bio. So as we keep going, I'll refresh you. The scientists cloned fragments of the CD3 gamma gene that included the first transcribed nucleotides plus up to 780 nucleotides of upstream regulatory sequences into plasmids in which the gene for the firefly enzyme luciferase immediately follows the fragments. The plasmids were then introduced into a line of T lymphocytes, which is in figure 1, and the cells were allowed to grow for a short while. Because the regulatory sequences of the CD3 gamma gene immediately precede, okay, so precede, that's pretty important, the luciferase gene in the plasmids, the activity, which is either positive or negative of the regulatory sequences, affected the amount of luciferase gene expression by the T lymphocytes. Okay, so that positive negative expression positive or negative effect of those sequences determine how much luciferase gene is expressed. So luciferase catalyzes a reaction that results in the release of light and is responsible for the bioluminescence or light flashes of fireflies. By quantifying the bioluminescence or luciferase activity in the cells, the scientists were able to determine the effects of each CD3 gamma gene fragment cloned into the plasmids on the expression of the gene. So if I look at this picture here, I could see right here in figure 1, so I have my upstream regulatory gene sequences and then my transcribed region. Okay, so it's very similar. Okay, so this is for a eukaryotic gene. Remember, you have that promoter region, and if that promoter region is bound to those transcription factors, that means that gene is going to get transcribed. Okay, so it's the same thing as what this problem is saying. Okay, so the only difference is that this is in a prokaryote, so this is in a plasmid. Remember our last FRQ, we talked about plasmids and how they're circular pieces of DNA. So you have that CD3 gamma sequence that's inserted right before that luciferase gene was inserted. And so if that CD3 gamma sequence is inserted, then that means that luciferase gene will be coded. All right, so figure 2. Figure 2 is crazy. Okay, so I'm going to go ahead. I'm going to actually start here at the bottom. Okay, so I have a parent plasmid with non-CD3 gamma active promoter. That means I have some sort of different promoter. And I can see this is where I have most of my luciferase activity. Okay, so this right here is going to be my positive control. Positive control means that all of my regulatory sequences are turned on. I'm going to have the most amount of luciferase coded. Right here with a parent plasmid, okay, because if we think about bacteria, if we think about a regular bacteria that's been inserted into a T lymphocyte, okay, they're not going to glow, okay, so they're going to have very little luciferase activity. So if I don't have that gene that's going to cause that luciferase to encode, I'm going to have very little luciferase activity. So right here, I have different types of plasmids. And those plasmids all have different types of genes. Remember, they said up to 789 nucleotides. And so because they have different genes, they're going to be producing varying amounts of luciferase in that T lymphocyte. All right, so hopefully that graph is a little bit easier now. Okay, so let's go ahead and start with part A. Identify both the cellular component, okay, and the location of that component that's responsible for producing the luciferase protein from mRNAs transcribed in the plasmid. Okay, so containing T lymphocytes. Okay, so that part's really easy. That has absolutely nothing to do with this experiment. Okay, so basically it's asking, okay, how do we get this luciferase protein? Okay, so where, okay, is luciferase, wow, goodness, luciferase protein encoded, okay, and basically which organelle does it? Okay, so if we think back to transcription translation, that's really easy. Okay, we're talking about the ribosomes. All right, so the second part though, the second part is what might get a little tricky. It says the location of that component. So normally you would think the rough ER, but if we go back up here in our reading, okay, so it says the signaling protein is only found in the plasma membrane of the immune system T lymphocytes. Okay, well, we know ribosomes are not in the plasma membrane, but they're in the cytosol, which is right next to the plasma membrane. So we can say that these ribosomes, because it's only found in the plasma membrane, they're accessing the ribosomes that are right there in the cytoplasm. Okay, so we know right here this location. Okay, so where is the luciferase protein encoded? It's going to be encoded in the cytoplasm, okay, by those ribosomes. So next, explain what dictates the lymphocytes the correct order in which amino acids should be linked to form the luciferase protein. Okay, so again, a really easy question. Okay, so what determines the order of amino acids? Okay, so if we think back to our transcription translation lesson, then we know that DNA, okay, our central dogma, DNA is going to code for mRNA, mRNA is going to code for those amino acids. And so that's all that question is asking for. So technically it's asking, let's see here, so what's the correct order for amino acids? So it's directly asking for the mRNA, but you would still get credit if you said that the DNA codes for the mRNA, so it transcribes for the mRNA, which codes for the amino acids. You would still get credit for that. All right, so let's go to part B. Okay, identify the independent variable in the experiment described. Okay, so independent variable, remember, is your manipulated variable. What is it that you are changing? So if you remember dry mix, okay, so dry mix, so dependent responding y-axis, manipulated independent x-axis. So if you remember that, that's great. Unfortunately, this graph is turned sideways. And so because this graph is turned sideways, we need to actually look at it the opposite direction. So instead of dependent responding being on the y-axis, that's actually on the x-axis. So our independent over here is going to be our plasmids. Okay, so our plasmids, those are the things we're manipulating. We tested several different plasmids. So that's going to be our independent variable. Okay, so independent. Okay, so what did we experiment with? Okay, all right, so that's going to bug me. There we go. All right, so identify the plasmid that was used as a negative control. Okay, so remember, a negative control means that you're not going to see much activity. Okay, so and I kind of went over that when I went over the graph. Okay, so I have all my different plasmids, but down here, there's my parent, which is my original, and a parent with a different promoter. So my parent plasmid by itself, that's going to be the one that doesn't have much activity. So we consider that a negative control. Okay, remember this one down here at the very bottom, that's my positive control because it has the most activity. Okay, so for a negative control, okay, that's my least activity. All right, so justify including the plasmid with a non-CD3 gamma active promoter in the experiments. Okay, well, we just talked about that. Okay, so a positive control ensures highest amount of activity. Okay, so, and we need to have that. We need to have both positive and negative controls. All right, so next, identify the plasmid that must contain the CD3 gamma core promoter sequence but the fewest or no negative regulatory sequences. Okay, so fewest or no regulatory sequences. So that means that we're going to be producing a lot of that activity. Okay, so based on the data in figure 2, describe the most likely cause of variation. Okay, we'll go to that in a second. All right, so if I look up here, okay, so which one up here, okay, has the most amount of activity, okay, but is one of our test plasmids. And we could see it's right here, this one that's dash 15. Dash 15 has a lot of luciferase activity. So that means that there's a lot of positive regulation and not very many negative regulations. Okay, so which one, okay, so which plasmid, okay, shows the most that's not a control? Okay. All right, so next, describe the most likely cause of the variation in luciferase activity among the cells that contain plasmids, and I'm just going to say the numbers. Plasmids 419, 309, 239, and 199. Okay, so if I look up here, okay, so it's these here right in the middle, and we can see they're not exactly the same. Okay, we have varying amounts as well as varying, I think these are standard error bars. Yeah, standard error bars. All right, so we have a varying amount of activity. So if we think about it, so all of these positive and negative regulations, so essentially it's kind of like putting, having, let's say, a traffic light. So let's say, for example, everybody has, they're all starting at the same starting line, but you have, different types of cars. So like, let's say somebody has a classic car, somebody has a sports car, somebody has a run-of-the-mill car, somebody has a truck. Okay, so on go, okay, people are going to be able to, go to the next light. Okay, so whenever a red traffic light hits, everybody has to stop. Doesn't matter where they're at. Okay, so like let's say that you have a really old broken down car, it's not going to be able to go as fast as, let's say, a sports car. Okay, so at that point, even though you've said go, because those stop signs, determine like, when that car has to stop, you're going to have cars at different lengths away from that starting line. Okay, so because of that, we have varying amounts of negative and positive regulations. Okay. So another example would be, like let's say in the classroom, let's say that first, I want to try to figure out the average height of everybody in the classroom. Okay, but let's say first I only do the sophomores, and next I only do the seniors. Okay, next I only do the boys. Next I only do the girls. Okay, so that's going to give me different averages, and it's not going to actually tell me the true average of everybody in the classroom. Okay? All right, so next, calculate the approximate percent increase in luciferase activity between cells containing plasmid 59 and plasmid 149. Round to the nearest whole number. So 59 and 149. So if I see right here, 59, okay, is about at 4. Let's see here. 149 is at 4, 59 is at 2. Okay, so there's a couple things you could say. You could say that it increased by 2. You could say that it doubled, or you could say it increased by 100%. Okay, that one was fairly easy. All right, part D. Predict the most likely observed level of luciferase activity if plasmid 789 is introduced into non-lymphoid cells such as kidney tissue cells, and justify your prediction. All right, so we're going to insert this into a kidney cell. Well, if I look back up here again, okay, so typically only found in the plasma membrane of immune system T lymphocytes. All right, so if it's only found in those cells, do you think it's going to work in a kidney cell? Probably not. All right, so, okay, would this work in a kidney cell? Okay, and if we look at this promoter region again, so the reason for that is because each of your cells has specific transcription factors. So those T lymphocytes have a transcription factor in order to encode that gene. Okay, that's what makes your cells different. They have different transcription factors to transcribe whatever it is that they need. So your kidney cells probably don't have that. So kidney cells have different transcription factors. Okay, all right. So I think we went over that. Hopefully I didn't run out of time, But if you have any other questions, definitely let me know. All right. So I hope you guys